Hi everyone, it's a pleasure to keep up the streak with the European useR, so it's nice to be here. Anyone have been already trying out this ISEE tools? Are there any early adopters in the audience? Yeah, awesome, cool, okay. So you can stay because there are new features that you probably might have not seen from the last time. So first of all, a few words about me. Who am I? I'm Federico, as you have heard, and I'm a postdoctoral fellow at the CTH Mainz at the IMBY. I could speak quite a lot to you about platelets, but for the interest of time, you can just write to me in any of these formats. And there's a QR code. You can get the link to the presentation. You can tweet about it, and we can keep on talking there. So to quickly brief, so thank you, Michael, for introducing a little about summarize experiment. Just a few words about what goes on in terms of RNA sequencing. If you take a high-dimensional snapshot of whatever goes on transcriptionally in a cell or in a sample for RNA species, then you get what is called RNA sequencing. It can get complex, but in the end, it all boils down to vector values for each sample and to, math processing error. That should be a times, but so it's I can go on with that. So it's a matrix in the end, but you can do a lot of things on this matrix. You can, for example, you can compare between conditions and try to pin down what are the molecular underpinnings of this. And if you go to the single cell level, which actually is a quite common technique that is used now, you can also, for example, you can discover cell subpopulations by the similarity of the profiles. You can check out their developmental trajectory and so on and so forth. Thing is hard to be done because the data is not big, or probably we might disagree on that. I call it large data. It's large data, especially RNA sequencing is quite heterogeneous, and it's also quite sparse. So you need proper tools to do that. You've probably been there to today's keynote speech from Joe Cheng, so I'm still jiggling from that. And if you still, I can levitate now. I've been working on these kind of things for the last couple of years, and so if you want, you can check some of the manuscript that I've been working on. So it's all there. Thing is that, as I said, you need proper tools to do this kind of visualization, and probably it's never been that important to have tools for exploring, visualizing your data, because you need these to do the following thing. You can have better quality checks on your data. You can generate also better research hypotheses. You can represent your results better, and you can communicate them better. So summarize experiment is an excellent, efficient container for doing that. In the end, it's quite a compact yet super powerful annotated table where you have, for example, features, something about your features, which might be your genes, and samples, which might be your samples. So the columns are in here. And especially this is true for single-cell datasets. You cannot think, sorry, Diana, you said you cannot think of more than 3D. You can get to 6D, but you cannot probably get to 10,000 dimensions. So you need some proper reduced dimensionality approaches to do that. And you can also store these in slots which are properly included in this single-cell experiment class, for example. And if you're into single-cell data, you should definitely check out this OSCA bioconductor.org, because we've been working on a kind of a book-down-based book that kind of tells you how to orchestrate single-cell analysis with Bioconductor. So there comes Marie Kondo, and she's asking you whether you are having joy in exploring your own data. And if you're not, like Marie Kondo says, you should throw away your tools. I'm not that drastical, but you should just at least try ISEE. We were looking for a silver bullet or a silver-coated bullet, and when I say we, I mean Aaron, Charlotte, and Kevin. So we got together after the Bioconductor conference in 2017 in Cambridge, and we found out that there was no general tool for doing this, and on top of it, there is no support for reproducibility. So we come around and said, okay, after a couple of brainstorming sessions for deciding the name, we said we could actually have an interactive summarize experiment explorer, and there came ISEE. So if you like the stickers, I have a few still left. You can come and grab it after that. Otherwise, there's a new batch coming, and I can send it to you per post as well. Otherwise, the package is available in Bioconductor, and the publication about that is in F1000 Research with nicely live apps included. So if you can write try before you buy. So you don't even need to install it. And we put some effort to make it as simple as possible to use it, and indeed, so with some simplification, so there might be some lines that will not be shown here to get to this object, but in the end, to do the entire exploration, you just need to do ISEE, SCE. And I'll just kind of make a showcase for that. You might have been heard about this tabula muris dataset, so it's a mouse atlas for expression for quite complex, so it's 20 organs and tissues from 8 different mice, 20,000-ish genes, 40,000-ish cells, so you get to 1 billion of data points, and how do you deal with that? We have been reconstructing one of the analyses at this repo for when I'm trying to showcase one of these IC instances. You can check it out later, but I will just give you a tour of the panels. So this is how it can look like when you start it. You can have, for example, a reduced dimension plot. You might be familiar with t-SNE plots. You can have feature assay plots, or whatever is on somehow gene related on the y-axis. You can have sample assay plots, so whatever is sample assay on the x or y-axis. You can do a lot of these things on this summarized experiments. Obviously, you can do, for example, you can have column data, so whatever you've been annotating on one side, and row data, and you can also have tables that you can, data table-like, you can just use the search, and then you can link all of this. This will come later. So you can have it like that, but this is what happens when people come to you and say, what do they say? Who recognizes the song? I want it that way. Exactly. So someone is into the 90s. That's good. Okay, so they come to me and say, I want it that way. They want flexibility. They want customizability. So how do we do that? For example, they might want to reorder the panels, and they can do it. So they can just select it back and move, and then just by clicking, they get this configuration, which is just what they wanted. Or you can do more. You can, for example, color each dot by augmenting the data with biological knowledge. That's how I know it. I call it. So you can take a feature name here. You can select a gene, which might be a marker of one of these samples, and then there you go. So PAX9 is there. You can link the panels all together, so it should, oh, oops, I'll just do an F5 because it doesn't look that nice, so I'll just hope. You can link all the panels to each other, and you can do that by the brushing mechanism in Shiny. So, oh, okay. Okay, you can do that. We are sticking. Okay, so for example, you can check what happens. So I'm telling that I want to pass this row statistic table to these element here. So whatever I select here then gets selected as a feature name. So my gene is going to be plotted just right there, and I will just have to click. So there's no need to type or to do something like that. You can link it to more of these plots. So you can see that this one is transmitting now to this feature assay plot and to this reduced dimension plot as well. So I can do many things at once and just pointing, clicking, no need to know how the code works. Nevertheless, you can also have custom panels because, say, it's for any kind of users. You can be a beginner or a wet lab scientist. Not saying that they're all beginners, but you can have some extra needs. And for these extra needs, we allow for custom data plots and custom statistic table. What you can do, we implemented, for example, a multiple selection, also with quite a clunky lasso freehand selection that I think Joe promised that they will be working on soon, probably after they're done with this Shiny meta. But you can do the following. You can select 2 different populations of cell over here and select a subset of these genes. What do we want to do? For example, we have different profiles in these 2 clusters, and what we get is just, say, a table over here and a heat map-like, will come now, representation of whatever logfold change between these two clusters, and it will be now. Come on. Okay. You can trust me on that. Okay, see. We said reproducibility. And how many times have you been staring at a nice plot in a publication and you ask how to draw this beautiful plot? So, and this is the reaction where I get from collaborators when I gave them the code. So I'm not saying it's boring, but it could be. So with ISEE, we provide a 2 clicks button, full access to the whole code, source code of whatever you've been seeing. So one-to-one, you can reproduce the exact things. And what you can do, you can just Control-A, Control-C, Control-V in your script, or even better, you can have a R Markdown. And Britney Spears is actually the most entitled singer for talking about reproducibility, because you can knit it maybe one more time. So, or oops, she did it again. So we just recompiled the report. So this is a situation that you might have been, I don't know, if you're not familiar with a user interface, it can be overwhelming. And we don't want this to happen to our users. So what did we do? Interactive tours based on this R-intro.js JavaScript library. And basically what we do is that we take the user through the user interface and kind of force him to do and to learn by doing what you can do. So this is just a quick one, but we did implement a couple of these tours to showcase what everything ISEE can do. And that's quite a cool thing, because you can use that also for deploying a fully fledged application. It's not just your publication, but it's also like empowering other users to check your data, if you're not scared of that. You shouldn't. We were actually delighted to see that I don't know why these are rendered in a strange way, but anyway, so we won the Shiny contest for the most technically impressive solution, probably because we have been working on something that Joe was struggling with for quite some time, but I'm happy for that he came probably with a better solution. Anyway, the next question would be, are we really probably reinventing the wheel in terms of expression browsers? I've been collecting a couple, and just not more than a couple, it's quite 40-ish or more of these gene expression browsers. And probably we should not be investing that much of development time to do yet another expression browser, because you can just use ISEE, or you can just at least give it a try. And this is just a funny feature. It started like that, but I can
show panel reduce dimension plot 1. Show panel heat map 1. Height panel reduce dimension plot 2.
So it works with X cracking in the background. So this was Kevin. This was Kevin. Oh, I don't know what have you been checking. Okay, this was Kevin after he implemented the first prototype of voice control for ISEE. So it's based on the Anyang library. I can give you the details later. But the cool thing, actually, it can be a super cool feature in terms of accessibility, because people might not be able to just use it like that. And there are a couple of Easter eggs. One of them is ISEE, and the other one is Game of Thrones based. So have fun looking for that. In summary, we give you a lot of flexibility, customizability. You can link and empower the user to have a better extraction of information. You can guide your users through the whole exploration. You can effectively communicate your results. You have fully reproducibility for yourself in 2 months, in 2 weeks, in 2 days, in 2 hours, and for others. You have fun and accessibility with voice control. And if you've got data, you can just accompany your publication as a live browser. In terms of Outlook, so what we have been working on is probably I call it the dream of ISEE.it, because we will be able to upload a data, assuming that it's not too big, and then probably work on some multi-omics approaches of visualization. So I'm a big fan of Douglas Adams and the Hitchhiker Guide to the Galaxy, so if I have a big long-term aim, I would like this quote, which is beautiful and very fitting for the exploration of data, to become ISEE first, think later, and then test, but always ISEE first. So I'm dreaming, okay. So there's tons of links, so you can go from, say, just using to, say, from 0 to useR in ISEE. You can see a lot of examples that we've been working on. Here is, again, the link to the presentation. Thanks a lot to my institutes that sponsored and, allowed me to work on this. And I'm just, 1/4 of the team. So without these guys, this thing will not be existing, and I'm super thankful and happy that I've got to work with them. And thank you for your attention, and I'm happy to take any question if we have time.
Yes. Thank you, Federico. We have time for questions. Three minutes, in fact. Yeah, there's Diane.
Hi, Diane.
How large a data set can you have?
Name a RAM limitation that you might have, and it can get large. So this tabula muris is quite large-ish. We didn't try with this 1.3 million neurons dataset from 10x Genomics yet, but we have been experiencing a little more to the upper end of, say, 100,000 cells times 20,000 job genes, which is quite a large one. And it takes a while to re-render all the plots, but it works. So you just need to provide a powerful computer enough to do that. And I think Thomas Pedersen is working on some kind of a speed up procedures for ggplot, so we will all benefit for that.
Any other question? I have one.
Yes, please.
Or did you just wanted to ask something?
No.
Okay. So it's a great example for like a collaborative project.
Okay.
If you could do, so it took you from 2017, right? It took you like 2 years.
No, 3 months.
3 months.
We had Aaron on our side, so that's kind of like cheating. But so it was a incredibly quick project from conception to realization. I hope that all my project will be like that, but it's.
Yeah, keep working with them.
Yeah, I will. We are struggling in our free time to just keep on doing something together. So yeah. It's a great team. So.
Okay, thank you.
Okay. Thanks.